Yeah.
Do you have power there, Bruce?
Huh?
Did you get power back yet?
We did. We got the power back on Saturday.
Okay.
And
That's
today
nice.
we had somebody come by that is looking at a fireplace insert that'll, make better use of the gas if this happens again.
Oh my God. We lost power for a while, so we lit a fire in the fireplace. Like produces no heat. I think it just creates like a
Suction.
suction to suck out the existing heat.
Some of the newer ones, the old ones. I've got a heatilator at the lake, and it works. It heats. I can feel it, but as soon as it goes out, it's cold and it starts sucking warm air up the chimney. So
Yep.
Yeah. And they don't have chimney flues for those things. I mean, can't shut it. So,
oh, right. Okay.
yeah. Back in the day when you make a fire, you'd open the flue.
Yep.
And then you close it.
And you had to remember to open the flue.
That's right. Sometimes you didn't. The other thing you don't want to do is to turn on the kitchen fan.
Oh yeah.
And run the fire. Yep. Well, let's see. Oh, there's Judy.
Yeah, but my name is wrong. I have to fix it. I've been hosting for someone else on another website.
Well, today John Wilsey is going to be our he's going to run the show. So, this is John. John, why don't you say hello to everybody?
Hey everyone.
That's the John Wilsey. He's he was probably the smartest guy at FLIR in many ways.
For a short period of time.
And I actually hired him. He's the only guy that got all the questions that I asked right, and he expounded upon them in ways that made me very happy that he came in the door.
Yeah.
And you were there for how many years?
16 years at FLIR.
Yeah. Yep.
What?
Hi, Tim.
Talking, but not the show. Sorry.
Hi there.
All right. Well,
How many people do we got? We got 18 per 18. Wow. Got that many? Huh.
it
Wow.
happens fast.
Well, it's 6:29 by my watch, so I'm gonna give it like another minute or two, and then I'll introduce you, Dan, and you can get going.
Okay. Thanks.
And I'm gonna just at some point, I think what I should do is just sign up to give another talk, and that'll like inspire me to actually prepare such talk.
Yeah.
Yeah, it has that effect, John.
Yeah, nothing like an imminent deadline to make you do some work.
It's called pressure.
It's called student syndrome.
Oh, all right. And I was on the other side. I was the teacher. And when I had to prepare a lecture, that put the pressure on me. I had to do it.
Yep.
Made a big difference.
Well, I have to say I've enjoyed the discussions and the discussions that follow the discussion. So one of the things I kind of feel I'm in isolation here is trying to find good books to read, and number of ideas that have come up have been pretty good. And finding something that was vetted by somebody else and can tell you which chapters to skip and things like that could be very valuable. So I'm wondering if we should at least start an online, discussion by emails to talk about good books you've read. Because a lot of.
I joined 2 book groups now that does it on Zoom, and so far, now it's not science. It's just general, wonderful reading books that people recommend, and it's really helped. Because then I don't need garbagey stuff. I'm reading stuff recommended.
Yeah. So if you want a good, like nerdy, sciencey book, I just read. So I say read, but it was actually on Audible, so I listened to it. But it was called Algorithms for Living, I think. And it was just, about different search algorithms, and it was fascinating. It sounds dorky, but it was really cool. So.
I think it would be cool for us to have some way of sharing things like this.
Yeah.
A way for us to communicate a little bit.
Bill Gates was on this evening. I was just listening to him a few minutes ago talking about his book. I should be getting that soon. How to avoid the climate change. And yeah, it was a good Zoom meeting, discussions.
Yeah, very cool. All right. Well, let's get going. So today's speaker is Dan Rossignol. So Daniel P. Rossignol has published over 50 scientific papers and holds over 75 patents. He has worked on both the discovery and development sides of the pharmaceutical industry for 28 years in the areas of sepsis, vaccines, oncology, neurology, and psychiatry. He has a BA in biology from the University of Connecticut, a PhD in biochemistry from the University of Illinois at the Medical Center, and postgraduate studies in glycobiology at Johns Hopkins University. After a 7-year stint in researching the mechanisms of action of pesticides, he decided his time was better spent in pharmaceutical development. He is now enjoying retirement, consulting, and trying to find time to hang out with his twin grandchildren. The title of his talk is Sepsis, A Tale of Woe. Sepsis, A Tale of Woe by Dan Rossignol.
Thank you, John. I'd also like to add to that I've got about 35 years experience in brewing beer too. So maybe we get an online discussion of that.
Hey, I've got like 20 years, so.
Yeah, it's changed a lot, hasn't it? It used to be a real niche thing, and now a lot of people are doing it and they're finding the advantages. So, okay, I'm going to turn on screen share. And what I'd like to say is that this kind of logically follows up the discussion from last, I guess a couple weeks ago now that I talked. And I'm going to see if I can bring this up here. How's that? Does that work? I'll go to presentation mode. Okay, can you see that?
No.
I'm not seeing it. No.
Not yet.
Not yet.
Did you push the green thing?
I thought I did. Share screen
Boy,
2.
you should say, push the share screen.
Okay, let me go back to let me escape here. All right, share screen. Select the window. And
Make sure you also click on the volume and the TV, the type of video you want. It's on the.
okay. Oh, there it is. Okay, share.
Yeah, you got it.
All righty there. Okay. So,
It's definitely not easy.
You see it now.
okay, good. So what I'm going to talk about is what first got me into pharmaceutical development. And I've since spent 28 years working on this one drug. I had the opportunity to both help design the molecule, optimize it, develop it through preclinical. And then when I was looking to move on, the clinical group offered me a position to go and help with clinical research. So those 3 phases of study, phase 1, 2, and 3, were more or less part of my life for, I think, an additional 10 years. And as I'll point out, unfortunately, at the end, the drug did not work in sepsis. And at the time, the kind of tongue-in-cheek joke about sepsis was that it killed 100,000 patients and 2 biotech companies a year. And ours turned out to be one of them. And we'll talk about it, and we'll talk about the logic that actually drove us to continue to do this. So we're going to talk about the science behind drug research, how drugs are approved, which we've more or less started to cover 2 weeks ago, in the role of the FDA. So this discussion is how a novel drug for severe sepsis was discovered and developed, and how clinical trials were performed, and we'll discuss the results. So some of the questions I will address either directly or offhandedly is where the structures of novel drugs come from, the role that animal research plays in drug research, and really why we need animal research. Can the process of drug development be sped up, and what could be the drawbacks? And that probably should be more discussion afterwards, because I really can't address all that here. And then the common phase 3 format of clinical trials. Why was it developed, and is there a better way to do it? And this has really become a burning issue in clinical research, because as I mentioned, EROM's law last time, that we are year by year, we have more and more failures and increased costs, and just trying to figure out a better way to do this thing. This whole process would be great, but you have to go in a safe manner, and you have to go in a scientifically logical manner. And if there was some way to shorten that, it would be great. So I mentioned that last time that we would focus more on early drug discovery and preclinical studies this time around. And we talked about clinical development and FDA review 2 weeks ago. But for early drug discovery, it's a complex process, and there are many ways to try to find drug structures. First, you have to have a target. Do we have a receptor? Do we have a neurotransmitter? What do we want to block or stimulate? And then when we get a hit, how do we discover these molecules? And there have been a number of ways to do that. And then, you also have to develop assays to figure out, is this a relevant assay for what I want? So I've got a molecule that inhibits something. Does that really mean anything towards drug development? Lately there's been a lot of discussion of high throughput screening, where you just take millions of compounds and you throw them at a receptor and say, does one of these work? And this has been a big push in the last 20 years. It's met with very limited success, but it seems to be something everybody's grabbing onto, because the old ways of making a molecule and testing it have just gotten too expensive. The process I'm going to show you is the old way of making a molecule and testing it. And then once you have something that works, you optimize it. And there's a number of targets that you have for that. So, and then once you have your molecule, you go both in vitro and in vivo and in vitro, and then ex vivo. You try to find ways to test the molecule and stress the conditions that the molecule might work. You want to make sure it's going to work. And then you do a thing called administration, distribution, metabolism, and excretion. So you got to know what the animal is doing with your drug. And once you look at all this, you have a pretty good idea what's going to happen in humans. If you take this drug and it immediately clears in these animals or never gets in the blood, that's pretty much what's going to happen in humans. And the distribution, if the drug only goes to the liver, then you're not going to treat kidney cancer with it, things like that. And metabolism, is it metabolized slowly? Is it metabolized fast? And when you metabolize it, is that going to screw up the metabolism of other drugs? That's an important concept when you've got a patient that's on a dozen other drugs. Suddenly they overdose on those other drugs because your molecule inhibits your body's ability to metabolize those drugs. So there's proof of concept. You've got a molecule, you're going to say it works. There's drug delivery. How are you going to get it into the person? Do we have to do IV? Can we do oral? There's formulation. You got to figure out your dose. And then you have to do your studies to enable you to do your investigational new drug application. We'll talk about that a little bit in the IND application. And then these were what we talked about a little bit, phase 1, 2, and 3, and how to test the drug through, hopefully to get it as an application for a new drug application. Does anybody have any questions or anything right on this? We're going to dig into all these things a little bit more. So I mentioned last time that you have drug discovery, preclinical, clinical trials. You have maybe 5 compounds, FDA review. And with any luck, you can get this whole thing done in less than 17 years. And I say 17 years because that's your patent lifetime. Once you patent a drug, you got 17 years to get it out there, sell it, and get your money back. And every year over 17 years is just wasted money. So the molecule I worked on mostly was in this area. We didn't screen 10,000 compounds. We worked kind of in this range. We had molecules that worked. We needed to make them work better. And I'm going to show you that. And then I'll talk about the clinical trials. So first off, lessons in the path to drug development. You know, things that, I don't know, in a nutshell, this is what the story of drug development is. Synthesizing your compound can be tough. The first time it's made, the first milligram of the drug that we actually developed cost $1 million to make. It was $1 million milligram. And I weighed it out very carefully, I can tell you that. After that, the compound synthesis gets better and better, and we got it down to somewhere around a few bucks a milligram once it finally went into the final clinical phases. Getting the drug into the body, how you're going to mix it and formulate it is actually an art. And if you don't do it right, the drug goes into your gut and doesn't get absorbed, or it gets immediately metabolized, things like that. Drug administration can be complex. If you have an IV drug, how often should you give it? Where should you give it? If you want to get it to the brain, do you have to put it right into the brain, you know, via the cerebrospinal fluid, or can you give it IV? Does it get across? All these things have to be dealt with prior to you actually deciding that this is your molecule. Pharmacological activity. Now that's what the drug does in the body. It can be confusing. You can even have a good assay for what your molecule is doing, and it can still be confusing. I mean, for instance, the statins, forever the statins were shown to inhibit cholesterol synthesis. But in reality, they think the way statins work is it causes the liver to increase its uptake of cholesterol. The ultimate goal, because it inhibits synthesis, the liver ends up taking up cholesterol and thereby reducing your LDL cholesterol. So the idea of how it actually works and finally is often, we don't know. We get these things out there, they work. Okay, I'm not really sure we know how this thing works. Just thank goodness it works, because we can sell it and treat people and make a few bucks. Clinical activity can be hard to measure. For instance, you know it works. In this case, I'm going to show you that we know the drug worked. It just didn't work on the disease and the people who were afflicted. And sometimes you've got to get just the right population of people. Sometimes you got to get just the right genetics. And these things can be very hard to work out. In fact, a drug can fail and you just never know why. So otherwise, besides all this, it's easy. As I mentioned, the whole process here is gathering data through the whole thing. And it's hardcore. Everything has to have a verifiable source. And everything you do has to be traced every step of the way through analysis. So when the FDA looks at your investigational new drug application, they look at your animal data or your in vitro data. They look at the individual numbers. And if they want, they can come back and say, I'd like to see the notebook where you did that experiment and see the numbers in your notebook. So everything has to be, and your notebooks are all supposed to be signed off. You sign every page of your notebook and data, and you're supposed to have a witness that signs every page. A lot of this may be mundane to a lot of guys who are used to working in patented areas, but all this stuff is hardcore. And everything can be reviewed by hardcore statisticians and data analysis experts at the FDA. So none of this stuff is just made up. You know, you hear about publications that are made up. Well, if you try to do that here, you're usually going to get tripped up. So how can a drug intervention be shown to be safe and effective? And I'm going to talk about the development of clinical trial to find the truth about safety and efficacy. And the drug I'm going to talk about is called eritoran, and it's also called E5564, which was the, drugs are usually given a number in development. And eritoran stands for Eisai Research Institute Toll-like Receptor 4 Antagonist. So there's going to be a quiz at the end, and I'm going to ask you to come up with that. but I worked at ASI Research Institute, which was a group at ASI, and toll-like receptor 4 is the target of the molecule, and it's an antagonist. So that's, this is part of the fun of scientists getting to sit around, drink beer, and come up with names for molecules. So the disease we decided we wanted to treat is severe sepsis. Now, sepsis is defined as suspected or microbiologically proven infection together with the systemic inflammatory response syndrome, or SIRS. And the syndrome is characterized by a loss of local control of inflammatory response, leading to a profound generalized systemic amplification of the inflammatory cascade. This is generally a 40% lethal syndrome when it gets this far. So severe sepsis is defined as sepsis plus sepsis-induced acute organ dysfunction or failure. Organs are hypoperfused, or you get hypotension, often leading to death. Like I said, it was about 40%. So it's not a fun disease. It's not something that you kind of, oh, I think I've got sepsis. We actually had a woman who was calling one of our medical doctors all the time from phone booths saying, I think I have severe sepsis. And he'd say, well, if you're calling me, then you don't. So what this is cytokine storm. And cytokine storm was defined for sepsis. You hear about it now for COVID and all that. And in fact, Erythrorin is now being tested in severe COVID-19 infection and sepsis. The cytokine storm is, again, was defined for sepsis and septic shock. So how does too robust an immune response kill us? So as an immune response increases, so do levels of cytokines and other immune modulators, triggering a cytokine storm. And then what happens is blood pressure drops. Plasma fluid extravasates, it leaves the blood vessels, even though the cardiac output increases and organ perfusion drops. Organs become hypoxic and fail, death can result. And in our trial, we had an algorithm for measuring organ dysfunction and organ failure. And it was difficult getting subjects into this study because they had to have a kidney dysfunction of a certain level, or a hepatic dysfunction of a certain level, or a pulmonary dysfunction. So we had subjects in this study that had blood pressures, systolic blood pressures of 0. So we ask, was something here an evolutionary benefit? I don't know, they talk about having the blood run away from the organs, essentially, the critical organs, perhaps because the blood had a toxin in it. And we don't know, but there is a reason why it's so severe and so acute. We haven't figured it out if there really is an evolutionary benefit or it's just the system run amok. So some of the triggers of inflammation are these toll-like receptors, and humans have 9 toll-like receptors. I won't go into detail about them, but they exist on immune cells, macrophages, and they recognize danger-associated molecular patterns, or DAMPs, or microbial products from gram-negative bacteria, gram-positive bacteria, or fungi, and among other things. And these are called PAMPs, pathogen-associated molecular patterns. And these aren't like antibodies where you get exposed to them and your body says, oh boy, I don't ever want to see this stuff again. And you go off and make antibodies. These are innate. This is called the innate immune system. You are born with the ability to respond to these things. Your toll-like receptors are in your immune system at very, very early ages. They found toll-like receptors in sponges. So these things go back evolutionarily a long time. They've been proven to work for some reason or other, but they are there, and you don't need to develop or acquire an immune response. You have this toll-like receptor response. So the endotoxin receptor, when we first started working on this drug, we were looking for an endotoxin receptor. It was later determined to be toll-like receptor 4. And that's involved in the pathogenesis of severe sepsis, almost for sure. Toll-like receptor 4 recognizes endotoxin and certain fragments from necrotic cells as well. So if tissues start to die, you get an inflammatory response. And you see this a lot of times when you have inflammation due to tissue injury, where you get TNF, tumor necrosis factor, and things like that, probably triggered through toll-like receptor 4. So anyway, gram-negative bacteria have this outer membrane. That's what makes them gram-negative. And in that outer membrane is this molecule called lipopolysaccharide or endotoxin. And that molecule has a long string of carbohydrates and then what we call the business end down here. And we're going to focus on that. So your body hates endotoxin. Endotoxin can activate 40, maybe 50% of a cell's genes. When you look at what a cell can express in terms of a response to something, endotoxin can activate 40% of a cell's genes. There's nothing in the world that activates a cell that dramatically. Endotoxemia is detectable in most patients with severe sepsis. They think the endotoxin could come either from the bacteria or possibly even from your own gut, which is loaded with endotoxin from all the gram-negative bacteria in your gut. In animals, endotoxin is a principal activator of inflammatory markers or cytokines. And in septic patients, the appearance of cytokines is consistent with timing of endotoxemia. So when administered in low doses to subjects, endotoxin, LPS, elicits signs of similar, although less severe, to those seen in severe sepsis. And you can actually safely challenge healthy volunteers with small amounts of endotoxin. And we're going to discuss that in a few moments. Yeah, you can pay these mostly college kids because they're the only ones who aren't smart enough to know better, and they'll do this for money. You can shoot them up with endotoxin. We'll talk about that. So here's the business end, and I'm not going to go into much detail. The chemists complain that I don't talk about enough chemistry, and the biologists complain that I talk too much. But this region at the end, so those carbohydrates I showed you, and then this business end, this is called the lipid A region or toxic region. And E. coli lipid A here, at a casual observation, you notice that it's a diglucosamine diphosphate hexaacylated molecule. So there's 6 fats hanging on here. And these fats are very specific in chain length. And we call this bad lipid A because this activates the heck out of cells. This drives cells crazy when they see this at incredibly low concentrations. That's one of the fascinating things about endotoxin, is how well it activates cells at nanogram concentrations. And that's a good thing. You want to know when an infection is there, and you want to know it early. So now, if you look at bad lipid A, here's the structure that I just pointed out. And now you can look at some good lipid A's. There's a lipid A from a bacteria called Rhodobacter capsulatus and Rhodobacter sphaeroides. And I'm going to come to the reason why I'm showing you all this stuff. There are some differences in the structure, which I will not dwell on, but I used to know these backwards and forwards, and we designed an antagonist based on these non-toxic lipid A's. Basically, these lipid A's not only don't activate cells, but they will inhibit a cell from responding to bad lipid A. So moving forward, we took one of the lipid A's. Well, I'd like to diverge a minute and digress a minute and say, so we've got these good lipid A's. Why don't we just grow them up in some bugs, purify them, and use those as an antagonist for endotoxin? Well, the issues with growing up a bunch of bacteria and purifying lipid A's, which are very complex and I want to call them slimy, soapy molecules, are tough to deal with, is they have to be pure. They have your formula. You have to have they have to be stable, and you have to be able to formulate these things. So you grow up these good bugs, and you start to purify out their lipid A. First off, you get vanishingly small amounts by the time you get it ultra pure, and then these things, a lot of weird capabilities, but the minute you add any sort of calcium or anything, they just crash out. and go away. So they're very difficult to deal with. So what we decided to do was develop a synthetic program that would make molecules similar to these good lipid A's. So our first studies were in vitro. We screened several hundred analogs of good lipid A, and these were all synthetic, made by the chemists across the building. And as we made them, we would test them and we'd say, okay, this one works a little better. A works a little better than B. Let's go back and work around A, and you keep optimizing. So we tested these analogs against the ability of endotoxin to elicit cytokines in cells and blood from both humans and mice, as well as guinea pigs, and you name it. Anything that looked like it might be a good assay to test these molecules, we tested it. Ultimately, we found out that human whole blood was about the best. We take fresh blood from guys who work down the hall. I've done this assay many times myself. I'm a guy who I make about 4,000 picograms per mil of TNF when you trigger endotoxin, when you add endotoxin to my blood. So I've been one of the standards on this assay. And what happens is you add our target molecule along with endotoxin to blood. You see what happens there is without any of our target, without any of our antagonist, we get a good response. We get all this TNF released. But then as you start to add the candidate molecule, you can see we can inhibit this, and we can get this down to 100% inhibition. Literally, the cells make no tumor necrosis factor when you give them 10 nanomole per liter of our drug. This is a low concentration. This is good. Pharmacologists love anything around 110 nanomolar. This is great. And so, we do this assay over and over. Everything's compared to one molecule or another. And E5564 eventually became Eritorian. And this was just one really potent molecule. We actually gave this to a guy in Korea, a guy named Dr. Park, and he was able to elucidate the structure of toll-like receptor 4 using Eritorian. And it was great because not only were we able to get the structure of our drug sitting in the receptor, but he couldn't get endotoxin to do this with toll-like receptor 4. Our molecule did it. It stabilized the molecule so he could crystallize it and get the crystal structure. And this was something that we're really quite proud of, and that we were able to move science forward. And you can see, here's our molecule. Remember the parent molecule had 6 side chains. This only has 4, but this one bends back on itself. It's really long and it sits in the pocket. And that may be the reason why this inhibits the activation of toll-like receptor 4, whereas LPS doesn't. It doesn't have that fold back. It bends the molecule a little bit differently. But this was, again, this is a pretty cool thing for us nerdy type of pharmacologists. Okay, so does it work in animal models? I showed you some in vitro, even crystal structure. Does it work in animals? And yes, it works against endotoxemia and against infection models, which were animal models of sepsis. And I kind of like to say we saved a lot of mouse, rat, and other animals' lives, and it appears safe. So here is one of the assays that we did. Here, if you inject mice with bacteria and then you give them nothing, we get 90% lethality. So almost 100% of the mice die. If you give them drug alone, we get about almost 80% lethality. So we save a little bit, a few mouse lives here. If you give them an antibiotic, this is like a amoxicillin type, you can see that they get about 60%. So you save, from 90 down to 60%. But if you give them both at the same time, an hour after they get infected, the lethality drops to 20%. So the combination of the antibiotic and the drug that blocks endotoxin saved a lot of mouse lives. And again, this assay was done over and over again. We're fairly confident that we knew what we're talking about here. And this was dose dependent, and it was also, again, dependent on the presence of antibiotic. Does anybody have any questions? I'm covering 10 years worth of work here in 5 minutes. So that's cool, right? But you just can't go jamming it into humans. So first you test it in mice for toxicity. You do low dose, high dose, repeat dose. You look at gross safety signs. You look at, are the mice still cruising around? Do they show neurological signs? Do they show any, do they lose their balance? And then you actually do histology of all the tissues. You got to look at every tissue that in the mouse that's relevant, from reproductive tissues. You look at lung tissue, liver, kidneys. You make sure that you don't see any gross histological changes. Does it cause organs to change in any way? And then you have to do it in a non-rodent species. The FDA requires that you can do it in mice and rats, but then you got to move to a non-rodent species. And typically that's either some sort of a primate or a dog. In vaccines, I believe it's, you also have to do like ferrets or something, or they typically do ferrets, nasty little things. One of the other things you have to do is test for the ability of the drug to cause cancer. Does it cause mutations in DNA? And then you have to test for the ability of the drug to screw up other drugs that the patients might be taking. So you can do all this in vitro. You can actually buy frozen human liver cells and you add a typical antibiotic or an antifungal, and you see if the ability of our drug to, you test the ability of our drug to screw up the metabolism of these other drugs. So even before you go into humans, you have a pretty good idea that you have a drug that's going to react appropriately and not cause anything crazy. If it's going to cause something crazy and you say, okay, well, you can give our drug to a septic patient, but you can't give them an antibiotic, you're kind of screwed there. So you have to know these things. The other thing is formulation. And one of the things we found out was that this drug, pure, 99+% pure, forms what we call a micelle. It forms a nanoparticle with about 40 drug molecules per micelle, 7 nanometers. It's a tiny, tiny thing. It's similar in size to serum albumin, and it's approximately spherical in shape. And this is a remarkable amount of work. Normally you don't talk about formulation, but the guys who did this really took a drug that was going to be unusable, and they figured out how to get this in this shape. And the interesting thing is, once you have this molecule, if you just add a whisper of calcium or magnesium, it falls out of solution and becomes soap scum. And I call it very expensive soap scum because it's no longer soluble, no longer usable. So this was very carefully controlled, and they did a remarkable job. And you can spend years developing a workable formulation. We had some really good people who did this. Okay, so now we're going to go to the clinic. As I mentioned before, the phase 1 is going to be science and safety. We're going to try to figure out the pharmacokinetics, basically what the drug does in the body. The pharmacodynamics are what activity the drug has in the body. And we're going to look at safety. And we're going to then phase 2, a little bit of stats and science. We're going to try to see if the drug has some efficacy. And it's called proof of concept, and it's usually called a go, no go decision. So you don't do a big enough study to tell you that the drug really works, but you do a big enough study to tell you that you think the drug might be working. And this is probably the Achilles heel of clinical studies, is the phase 2. Nobody wants to spend a lot of money on a drug that isn't going to work, but then they don't want to spend a lot of money. They don't want to spend a lot of time and money on a drug that isn't going to work, but they got to spend enough to have a good feeling. And feelings aren't always good enough. Donald Trump can live with his gut feeling, but the rest of the world can't. So phase 3 now is stats rule. This is where your study has to be large enough to really get a good feel for safety and efficacy. And we have to establish clinically significant activity that'll allow us to file a marketing application with regulatory agencies. And a marketing application is called an NDA, or a new drug application. For a biological, it's called a BLA. And this is the key. The phase 3 is the key. And the design of the phase 3 is absolutely critical to making sure that your drug gets its best chance to work. So why are drugs so darn expensive? I'm just going to digress for a moment here. So preclinical, all that animal work, all that research, it only costs 2 to 10 million. Phase 1 to 3 can cost anywhere from 100 to 800 million. And after all that, more than 4 out of 5 drugs fail. So here you are now with 4 drugs that failed, cost you, 3.2 or $3 billion. You finally get one that works, you got to make up all that money. And it's just a very difficult thing to do. And people who think that pharmaceutical companies are out to grab money, I think all they really want to do is return on investment. And, if you're looking at developing a piece of software and trying to get somebody to invest in that, and, you got a guy like, some rich dude out there says, I want to invest some money. Drugs, just the return on investment's tough. It's real tough. So that is to $1 billion. It's just crazy. And in order to get past this, is there something that you can do to convince yourself that you're not going to waste money? And this is where I'm going to bring in some science that I think I'm real proud of. So first off, how do you start? You find a dose that causes only mild or moderate transient effects in animals. It's called the NOEL effect, the NOEL dose. So it's the no adverse effect level. And so you say, okay, it's safe in animals. If you give it to them for 7 days at this dose, you start at 1/10 of that in humans. And you look for those same effects. So say in mice, you saw that their liver function changed a little bit. You look very carefully in humans to make sure that same thing isn't happening. And then you slowly start to escalate the dose as you find it safe. So then you make sure it's safe as a single dose as you elevate the dose, as you escalate it. And then you do multiple doses and infusions. And the bottom line in our phase 1 studies is that we found minor effects that were mitigated. So a little bit of irritation of the vein and things like that. You dilute the drug down a little bit, and we were able to mitigate pretty much anything that we found. The drug looked very safe, and everything said, let's go. So now it's safe. How do we know it's working? And so you go down to New Orleans, and there's a center down there where they have volunteers that have repeatedly gotten dosed with endotoxin. And it's a low dose. It's about 4 nanograms per kilogram. And we did a double-blind, randomized, placebo-controlled study of our drug at very low doses. And we knew these doses based on our in vitro work. We had an idea how much drug we needed in the blood to work. And we gave them a half-hour infusion of either 50, 100, or 250 micrograms of drug. And then in the middle of that infusion, we gave them a shot of endotoxin, 4 nanograms per kilogram. And this amount of endotoxin is enough to give your average volunteer the flu for 4 hours. And to be blunt about it, they just feel like shit for 4 hours, and then they're really not normal for about a week. They still feel not so good, and then they recover and they come back in for another $800 study. Anyway, we gave them the drug, and what happened is that I'll come to that. So we say, okay, is that model where you inject endotoxin into humans, is that relevant? So we can do this. So what? Sepsis has endotoxemia, and of course, endotoxemia in healthy subjects has endotoxemia. The effects are mediated by endotoxin, likely in sepsis, and yes, in endotoxin, we know. Systemic inflammatory response syndrome is present, yes. Cytokine activation, yes. Short-term leukopenia, yes. Increased C-reactive protein, yes. Organ failure in sepsis, and some failure in some patients, yes. Fortunately, in our endotoxemia model, no, there's no organ failure. So what happened? If you look at interleukin-6, which is a pro-inflammatory cytokine, you look at over time, what you have here is, this is the placebo. I have this marked wrong. What happens is after about 1 hour, interleukin-6 plasma levels jump to about 2,300 picograms per mil and then drop. So after about 6 hours, it's kind of over. Now, when you look down here, these are the curves for 250, 150 micrograms. So here's 50 micrograms. Here's 100 in here, and buried in here is the 250 microgram dose. So the drug almost, it's extraordinarily well inhibited the response to endotoxin. And the same thing that happened with IL-6, we also found with things like tumor necrosis factor and other cytokines. In fact, in one case, the study site, when they randomized the groups, the first 4 subjects had drug, and they didn't know that because it was blinded, randomized. When the subjects were all sitting around after an hour or so after endotoxin, they thought that they had misdosed the endotoxin. They went in their office and recalculated, and they said, no, we're right. And the investigator said, this is the only time we have ever seen 0 response to endotoxin. And it was because they had what they did was they split the group into 2, and they grabbed out of the randomization, the first 4 had drug on board. And they said that this never happened before. So then, of course, the next group, it was randomized 6 treated and 2 placebo. The next group had, the next group of 4 had 2 and 2. And they says, oh, yeah, we were right all along. Yeah, well, so were we. All right, so just briefly, Erythrorin works. So it's besides inhibiting cytokines, Erythrorin inhibited all measured activities expected to be directly and indirectly triggered by endotoxin. Fever was knocked out, changes in heart rate, changes in whole blood cells, white blood cells, leukopenia, and then all the cytokines that I showed you, which is actually, to me, a little more dramatic. So we move from phase 1, where we're working in healthy volunteers. Now we go into patients with severe sepsis and septic shock. And the subjects that were chosen for the study, we tested the drug in about 200 patients in the United States. They had to be in early severe sepsis. So this means that, these aren't patients that were lingering in the ICU that had, sepsis days for days and days. They were in organ failure, the whole bit. We had to see these guys come in and basically have signs of severe sepsis in the ICU and be charted. And once they say, okay, look, this guy's got it, we can give him the drug. So early severe sepsis, they had to have at least 1 organ failure. And we measured changes in respiration, kidney function, liver function, and survival. And the gold standard for the FDA is what's called 28-day all-cause mortality. So if these subjects got really good, they got really well, and went home and got hit by a bus on their way home, they were considered dead in the study, 28-day all-cause mortality. That doesn't happen often, but it could, and it's something you got to think about. So you want to have your study powered so that odds and ends, small things like that, don't make a difference. But 28-day all-cause mortality is what the FDA likes. If you say, okay, let's get them off of the respirator, FDA goes, well, that's kind of subjective. Sometimes sites will leave you on the respirator longer. Sometimes they don't. So our kidney function, what is real kidney failure? It depends on age. FDA says, let's count mortality. That's easy to measure, and that's what we want. So the study was blinded, and I like to briefly review why blinding is important. If the hospital staff knows that a subject is on a hot new drug, what do you think happens? And this actually happened years ago with the drug. It was an antibody, and it was an unblinded study. There was only 1 treatment arm, and the subjects responded remarkably well. The drug was amazing. This was basically kicking butt on Wall Street. People were going crazy over this. This was the darling of Wall Street at the time. The company then turned around and did a phase 3 blinded study, and it didn't work. So what happens is when you've got a subject who's got sepsis, all that takes to improve survival is just a little bit more care. If you had a nurse sitting bedside, 100% of the time, your chances for survival are much better than your average hospital patient. That's just the way it is. You know, you don't have to wait to discover that, oh, I've got a new infection, a new fever, if the attention was greater. That is just the way it is. So without blinding, what you've got is a hospital staff that's really likes this drug. They're watching the patient like crazy. They're going to do better. Placebo. Why is it that, and what do you need? And I'm going to talk about this a little bit at the end, but a study is only as good as its placebo. If the placebo looks a little bit different than the active drug, the hospital staff will figure it out. And placebo is everything but the active drug. So as much as possible, a placebo arm of the study has to be as similar as the active arm. If there is a medication, it must look exactly like the treatment itself and be packaged identically. And the subject getting the placebo should not know that it is placebo. The person administering the placebo should not know it is active drug or placebo, double-blinded study. And the reason is, I'm going to come back. Hopefully we'll have the time for this. I'm almost there. So what happened in phase 2 study? You run a thing called a Kaplan-Meier curve. It's survival in the population that's called intent to treat. So this is everybody who signed up for the study and got in. Even if they only got one dose of drug, they were part of the intent to treat population. So on day 1, day 0, all groups had a 100% survival. But then you can start to see that, oh, okay, survival's starting to drop off here. And by 28 days, you can see that the placebo group and the low dose group had less survival than the high dose group. The study wasn't big enough to get a decent statistical significance, didn't have a p-value. But this, for our phase 2, was enough to say, let's continue on. Investigators were excited about it. We were excited about it. Everything looked pretty good. We got a difference here from placebo. So what are you going to do? You're going to continue on the low dose or the high dose? We went forward with the high dose. It was about 250 milligrams. So the phase 3 study is called the access study, and it's a controlled comparison of Erythrorin and severe sepsis. And one of the toughest things to do in clinical research is come up with a good name for a phase 3 study. And if you go to clinicaltrials.gov, you'll look at the names of these studies and they're all over the place. Some of them make no sense. This one actually is an acronym of what we did. 2,000 subjects were enrolled, this time around the world. We had patients in Japan, Italy, all around the world. The phase 2 study design, the study design was almost identical to that of the phase 2 study. And that's a good thing, because as soon as you start changing things, you have no idea what you got. Treatment had to be within 12 hours of organ failure. And I'm just going to say this is hard to do. We had hospitals, we had nurses crying on the phone because we think the organ failure 13, 14, 20 hours out, but I think this patient can use the drug. Sorry, they can't be on the study. The study was also randomized. In order to kind of beef up enrollment, we gave, for every subject that got placebo, we gave 2 subjects treatment. And this, in a way, is a little bit kind of psychological in that rarely does the patient alone sign the consent form for this study. This had to be next of kin. It had to be a caretaker. These patients were so sick that, you know, they're out of it. They're in the ICU. Many of them are intubated. We got the same thing going on now with COVID. You have to get next of kin to sign. And telling that next of kin that, okay, you got 2 out of 3 chances of getting the drug, because everybody thinks the drug is going to work. But you got to have placebo there to know if the drug is safe, you know, so you got a comparison group for safety. But you say, okay, you got a 2 out of 3 chance. Anyway, it was a tough study to do. It took years to do. And we finally completed enrollment at 1,984, 1984, somewhat Orwellian. And what happened? The
Hey,
treatment.
Dan,
Yeah.
real quick, time check. We're about 50 minutes in. 50.
Okay. I just got a few slides left.
Sounds good.
Good. Yeah, so the treatment did not reach its primary endpoint of a statistically significant reduction in 28-day all-cause mortality. This happened around New Year's Day. We got the data. And you knew it was going to be a bad year when you saw that there was no statistically significant difference. One of these things, you spend 20 years working on something and then it's like, oh no. So shop closed until further notice. Eisai at the same time lost the patent on another drug and basically closed shop. So next question is, how did this happen? What happened? And next couple slides, I only have like 3 slides left. What happened while this study was ongoing was that a new protocol for treating sepsis patients called early goal-directed therapy was introduced. And a guy named Manny Rivers, a great guy, you call him up on the phone, he'll talk to you. I really like this guy, but he killed my study. He worked at Henry Ford Hospital. This guy actually like slept in the intern's dormitory so he could do his work. He did a groundbreaking set of interventions in sepsis patients, and he did this all at the same time. So he would find patients early in sepsis. He would treat them aggressively early with antibiotics. He would treat them with lots of fluids and even blood transfusions if necessary. This treatment with fluids was completely against normal protocols. What you do normally is they would try to dry these patients out. He says, no, I'm going to pump these guys up. I'm going to get their blood pressure up and I'm going to get their oxygenation up. So he got their cardiac output up. In general, while this was being published and rolled out, most docs hated the idea because Manny changed so many variables at one time. They said, this isn't scientific. You can't do this. But they started implementing parts of this process because it made sense. So you talk to these docs at these meetings and they go, I'm not going to do all of this, but I'm going to pump up fluids, or I'm going to give antibiotics faster. I'm going to do all this stuff. Not all at the same time. And they, in fact, initiated a number of studies where they would try one of these things. They would try earlier antibiotics. Oh, that works a little bit. And then they would try lots of fluids. Oh, that works. And they put the two together and go, oh, that works even better. So basically, Manny broke the mold by trying all these things, and he was right. So during the course of my study, mortality due to sepsis dropped from normal 40% to about 24%. This was what it's going to be. I'm going to show you our placebo response was 24% death, which this is great news for patients, but it's bad news for me and Eisai. And you think about what would have happened if this study, so I'm going to go over why this was bad news for us, but you think about what would have happened if this study was not placebo controlled. So we had a historical mortality of 40%. We give our drug. If there was no placebo there, suddenly we got 24% mortality. That is what they call a relative reduction in mortality of about 40%. This would be phenomenal. I mean, I'd be a rich guy right now, but it was placebo controlled and we could see something else going on with these subjects. So this is what happens. So you have 40% mortality. If we could save 15% of them from death, you got 100 patients enrolled, 40 die in placebo, only 34 die in the treatment. When you have 24% mortality and you save 15% of them from death, 24 die in placebo and only 21.4 die in the treatment. This difference, this 15% difference is small. Your ability to see that signal drops. And that's indicated by here. So these things called power calculations. If you want to have an 80% chance of measuring this difference, I don't want to get too wonky on you here, but if you want a p-value of 0.05 and an 80% chance of actually seeing it, you need 2,000 patients, which is just what we had in our study. We had about 2,000 patients. But now at 24% mortality, with this small difference, you need 8,000 patients in your study. And what happened is our ability to see the signal was lost. So even if the drug worked, the study was not large enough to see it. And Eisai, after doing a $300 million study, Eisai didn't quite have the desire to go back and do something that was 4 times the size of that study on a drug that really was nearing the end of its patent. So now the drug is being tested in COVID patients. There are a number of independent investigators. We're giving the drug away and hoping that it will work. There's some evidence that the drug even works in viral infections, because we think there are pattern-associated molecules involved in sepsis due to virus. So anyway, that's thanks for your attention, and I'll take any questions.
Thanks, Dan. Interesting talk. Does anyone have any questions for Dan?
And did you look at any particular subgroups that might have been more likely to respond to your therapy?
Did you read that paper? Did you read the JAMA paper? I'll have to send it to you. I'm going to try to stop sharing here. We actually did. We found in one subgroup of patients, we measured endotoxin. And we found that patients who had no endotoxin in their blood, or if we gave the drug before they had endotoxin, there was a signal that they survived considerably better. But it was a small group. And unfortunately, that didn't translate out to the whole group. Otherwise, we would have seen a decrease in mortality. But the idea that the drug has to be around before endotoxin, we said, let's measure endotoxin in the blood. And a lot of septic patients either don't have endotoxin or it's transient, it comes and goes. At the time we gave them drug, the ones who didn't have endotoxin did better. But otherwise, we analyzed everything from hair color, eye color, number of middle initials. Nothing made a difference. Nothing made a difference. It was brutal. And these are the kinds of, when you got almost 2,000 subjects, you can do a lot of cross analyses, and we couldn't find anything.
Do the COVID studies include the use of extra fluids?
I don't know what the standard treatment is, but currently the treatment for sepsis and septic shock is to give fluids. They give lots of saline, basically. And I would say that if you've got a patient in septic shock for any reason, fluids are going to be the go-to. Going to be antibiotics or antivirals and fluids.
I was just thinking about the influence of fluids plus the drug. And it would seem like maybe that would potentially increase the impact like it did in the other study by the other doctor in Michigan, whatever.
Yes, yeah. Well, our drug was on top of, we didn't see any difference in early goal-directed therapy implementation in our group versus placebo. So it was pretty uniformly administered across both groups, and our drug was on top of that.
Oh, thank you.
So yeah, we didn't, it was blinded. So whatever the hospital does is standard procedures. And we had some restrictions on what kind of craziness they could do with these subjects. The antibiotics had to make sense. They either had to do a broad spectrum at first and then get a culture and go in with appropriate antibiotic, things like that. So everything had to be scientifically driven, and the sites were carefully chosen to do that. This wasn't your local community hospital. These are all university hospitals and medical centers.
I thought your study was really after the fluid study, where that was a known quantity to enhance fluids.
It was, but not everybody was implementing it when we started our study. We were right at that point. And in fact, when I called Manny up, I said, do you think a lot of places are going to use this? Should we change our power calculations? He goes, it's being brought up real slow. And my boss says, no, it's going to take 10 years for them to do that. Well, the hospitals did it. They rolled that. They saw that reduction in mortality. They said, we're going to do this. And it only makes sense. I mean, saline's cheap. You pump them up with saline and antibiotics. You start with that. So we underestimated how many hospitals would be using that therapy. And there's not much you could do. You can't sit on the drug and say, oh, let's wait till they all figure out if they're going to use early goal-directed. You got to keep going. And, our analyses during the study said that the drug is safe. So we didn't have any reason to stop. And it's just a brutal game of, expensive studies.
I just can't imagine you must have been so excited, at that point just before the very end.
Yep.
You must have been just extremely excited. And then the crash.
We would go to meetings, and I would have like the study nurses that actually dealt with the patients, and they would tell me, I got one that was treated. They'd say, I got one that's treated. I know he was treated. He was on death's doorstep at night, and the next morning he's sitting in bed reading the paper. And I says, write down his number. Tell me which number it is. We'll write that down, and when we unblind it, I can tell you whether he was treated or not. And it was up and down, I mean, sometimes septic patients just get better quickly, that sometimes they're just too dry. But after the study, it broke my heart, but I had doctors telling me that was the best run sepsis study they have ever done. I had other endotoxin people saying, we're not going to pursue endotoxin anymore because you prove that endotoxin isn't involved. Hey, great. Thank you very much. So, yeah, to have run the study and felt that it was as good as you could get. We didn't cut any corners. We just got hit with what it is. And the drug may work, it may not, but you reach a point where business decisions are made.
So Dan, the protocol was changed. How did they know that? Was there separate studies where they did all these treatment protocols and the mortality went down? So how was that separated from your drug study to whatever Dr. Manny did?
We actually specified in our study that a certain amount of fluid had to be given because we couldn't tell them not to do that. And then, antibiotics. So it was what the site normally does. It was site protocol plus our drug. And our study did specify certain things. Yeah, we want so many, okay. One of our definitions of shock, septic shock, was that shock was refractory to a bolus infusion of salt solution. So if you had a little old lady come in with a urinary tract infection and she was in shock, and you gave her a bolus of saline and an antibiotic, and she sat up and goes, oh, I'm good, I'm going home, I'm going to miss my soap opera or something. Those subjects could not be put on study. So we had to give them a fluid challenge before they could even be on the study. And it still had to all be done within 12 hours. So this is why the protocol was brutal. So there were certain things that we says, okay, we're not going to take a subject who just needs a little bit of salt water. We challenge them to a shock protocol, a shock recovery protocol.
You certainly got my attention when you said anyone that has a nurse 24/7 at your bedside, you will improve. That's quite the shocker.
Yeah, there's a little known study that showed that if you're very ill in a hospital and you're on a study and you're on placebo, you have a 30% improvement in survival.
Mm-hmm.
Just being on placebo in a study, because a study requires
placebo.
Placebos were great, but it's all the other treatment required under the protocol. Mm-hmm. It's the fact that we need to get liver function tests a lot more when you're on a protocol, and you need more physicals, you need more blood pressures, more EKGs, all these things that you do on protocol improve survival above the average care. So being on placebo in a study and having improved mortality, what does that say about standard of care? It's scary. I don't want to go there. Okay. I just,
It may also be a selection factor, Dan. It's patients who get into the studies tend to be better educated patients, more compliant patients, more likely to have family at the bedside who are monitoring what's going on.
You're probably right. I don't know if that study dove into that, but yeah. And you're right. They tend to be less complicated too. Oh, that was Mark, right? Who asked that question?
Right, yeah.
Yeah, you can't have a patient that's got 14 different organ failures and then, still get on your study for diabetes. It's,
There's been study done about placebos though, because I'm a nurse and I remember I worked a lot with patients on placebos, which we gave them to reduce pain. There were many reasons where we thought that they were just chronic complainers. And
Right.
so psychologically, if they think you've given them a miracle drug, they feel better, even though you just gave them glucose.
Right.
They feel better. And there's been a lot of research done on that too, the potent reactions to placebos.
Right.
Yeah,
I have another question for you. What would you suggest should be done with this whole trial process to make it efficient and cheaper and give you guidelines that make sense?
I've thought a lot about this and I consult with companies on design, and anything you can do in your phase 1 or preclinical to get you a dose. One of the toughest things, so in our sepsis study, we had a dose. Could you imagine if we had 2 doses because we didn't know which one worked? So it would add another third at least, So whatever you can do to know that you're getting receptor occupancy or whatever you need to optimize that phase 3. But I don't know how, you can combine phase 2 and 3, like kind of roll your data in, roll those subjects into another study and just keep beefing up your numbers. The FDA is allowing that sometimes, but they wouldn't allow us to do it. And, there's tricks, but nothing is groundbreaking to say, let's, for instance, the latest vaccine thing was groundbreaking because the mRNA didn't go through all the usual protein stuff and all that. That was all done at the basic research end, the science end. And then they really pushed through the clinical, and it was remarkable how they did that. Without COVID, it would have taken 5 years to get that vaccine approved. But under normal circumstances, you got to slog through the phase 2 and 3. So maybe we've learned a lesson here, but on the other hand, if you rush it through and it turns out to be unsafe, the worst thing you can do is get a drug approved for marketing, manufacture massive amounts of it, do your introduction to the clinic, get it all out there, and then see the toxicity. Like, Vioxx, I mean, that's just a disaster. It's a very expensive disaster. And, that's the last thing you want to have. So the process has its own checks and balances.
Mm-hmm.
All right, let me take a minute to introduce next week's speaker. So next week is Harold Hastings, correct, Bruce?
Yeah.
And the topic is Taming the Monster, Mathematics of Fractals in Art. So that sounds cool. I love fractals.
In art and science. Let me say, if anyone has children that have been coming here, have them come with pencils and paper and stuff, because in the beginning, I'm going to have people construct fractals on their own for the first 10 minutes or so. So,
all right.
So come.
Awesome.
And yeah, and Dan, I'm just wondering, I mean, with the COVID scenario, whether part of what lets you do it is, I mean, I know as someone with a couple of comorbidities and getting, as the Brits would say, COVID jab 2 tomorrow, that early on you could do a calculation, say, if this is the risk, it's still a benefit to me. So, I mean, for the first group, you were actually able to gather data on people who would benefit, even if the safety wasn't absolutely proven, because it was clearly of benefit to them.
Right. Yeah, I've actually, we had a neurology drug for Alzheimer's, and the FDA kept talking about carcinogenicity. And, you know, Alzheimer's treatment generally goes for up to 4 or 5 years, and it's at the end of life for the most part. This is why is the FDA concerned about whether it causes cancer 30 years down the road? And it's just part of the standard checking of boxes. And I guess, you know, the drug, if it works in Alzheimer's, maybe they'll move it up and do Parkinson's for 20 or 30 years. It's, you know, there's a lot of thought involved, and usually they think things through, but some of it's silly. And, you know,
Is there any way to allow patients to have a choice to take the risks they're willing to take in order to have the chance that some drug would work?
the patients have to be consented to a study like this. You explain the upsides and the downsides and the options.
I'm speaking about patients after a drug like yours has not been approved for release. If patients knew about it, could it be possible that FDA would allow patients to receive a drug like that, as long as it's understood the patient has decided, he understands there's a serious risk it won't work, but he wants to take that chance?
No.
Outside of clinical trials, doctors have the option of using any drug they want for any therapy, any illness they want. But of course, they don't want to use anything that's dangerous.
Okay, they have the option.
But it has to be approved, Mark. It's an approved drug written off label. Ours was not approved. So
you can still have compassionate use of drugs for extreme circumstances if there's no other hope allowed by any known therapy. Doctors can do a lot of stuff, including going to drug companies and saying, hey, we hear you have this drug that, you know, hasn't passed scrutiny yet, but we've got this patient who's going to die in the next 6 hours. We want to try it.
Yeah, your key word there is yet. So I worked on the compassionate use program for this drug, and it was mainly if it worked, and the FDA has taken 18 months to approve it. We had a compassionate use program to get it out there to doctors to use it because we felt it worked, but because we never even submitted it to the FDA. And, you know, the FDA hasn't had a chance to review the quality packaging, the quality manufacturing and things like that. I don't think there was much chance. Now, for COVID, because we did get the safety data, and we have, I think, reasonable data to support purity and stability and all that, the FDA was pretty, I understand, was pretty easy on allowing us into this huge trial, trying 20 or 30 different drugs in COVID. And it's going to be a mess because some of these subjects have gotten several experimental drugs, and trying to figure out which one actually worked for them. We'll have to see. But yeah, the drug is being put into a number of patients with COVID.
You know, the other thing is not only can you ask to use a drug, but some drugs are being used for other things. And it is not an official use that was part of the studies. I remember very well when I was working, and we would take pills, and I can't remember which ones, snap it open, put it under the patient's tongue, and use it for something entirely different.
Yeah.
That had nothing to do with what the drug was developed for. And I know probably that was not kosher, but I remember that happening fairly frequently when I was working. I worked in an ICU, and we would try anything, you know.
Yeah, the FDA has flat out said we can't control what doctors do. We can approve a drug for an indication. And in fact, some of the choices were this drug that we worked on, people talked about it being applicable to things like preterm birth, different kinds of, you know, infusion injuries, things like that. And the idea was if you could get it approved for a very small niche market, then the doctors might grab it up and use it for sepsis. And ethically, that wasn't a tenable thing for the company, but it is something that sometimes people will do. You could find, you know, okay, we got this market for 200 patients in the United States per year. Let's get it approved for that. So the FDA has now approved it and labeled it. And now the doctors can use it off label for sepsis and things like that. And that's not, you know, again, maybe some companies will do that. We did not choose to do that.
My question is it thinkable that the FDA would ever say, we have this drug, it comes in two versions. This drug that's out now has version 2, there is risk. You have to understand that. If you treat your patients, they have to understand only a 50-50 chance it will save their life, but it might. Was the FDA ever going to allow anything like that where it's understood there's risk, but the doctor and the patient can decide?
If, you know, they're talking about that now for the next generation vaccines. They're saying that they're going to be a little less rigorous on the early review for early, especially for early development. And, you know, basically they've got a platform. It was proven safe. At least we believe it's safe so far. And they may modify that a little bit. And the FDA is going to say, you know, go for it. Begin your phase 2 studies. Don't wait around and take another 6 months to get to phase 2. So get out there and, you know, determine if you get an antibody response. So we have so much data now on the antibody response and protection. All you got to do now is tinker with that a little bit and get the same antibody response, and we may be able to predict protection in humans. So it's kind of an exciting place to be for the people with the mRNA platforms right now. And the FDA has even said in this crisis, you know, with these variants, it might get even easier for you guys, which is exciting in one way, but in another way, you know what? One of these times we're going to cross antigens with some sort of cell protein, and you're going to raise antibodies to your own liver or something. We'll see. You got to know that that's not going to happen.
So is this common that, so your drug that you worked for 17 years on, that it gets kind of shelved, and like in a pandemic like COVID, it gets taken off the shelf and looked upon. So all the data that's there doesn't really go to waste. It's in this database that you referred to the last time.
Yes, yeah, the data is all been formatted for FDA submission and everything. And, you know, to me, my hopes were that they would find another use for this drug as we understand more and more about toll-like receptors. Toll-like receptors were discovered about the 5th year of my research. Forever, I did endotoxin receptor binding assays, and suddenly I had to change the name to toll-like receptor 4 binding assays. So we really don't know a lot about innate immunity and toll-like receptor 4, and I still hold out hope that, you know, we hold the world's best toll-like receptor 4 antagonist. And, you know, there are now hundreds of publications on E5564 and Eritoran for blocking this and blocking that, and including things like stroke, reperfusion injury in the brain, and things like that that are an inflammatory event.
I'm thinking of there are several cases where children have had later effects, cytokine storms that happened to them, and some died. And a drug like yours, given early on, may just have a chance to prevent that.
It would be nice. We never did children's studies. That was actually another thing that I was designing. You know, as we were getting ready to roll in the phase 3 data and submit the package, we had to decide how we were going to do children's studies, how we were going to do compassionate use, what the next disease target was going to be. There are so many things that we were going to do, and then it all just blew up. But I was working with these pediatric doctors at Cincinnati Children's, and they were very excited about the possibility of getting this drug into kids, because kids have a type of sepsis, like you say. If you could just block that cytokine storm, they are so healthy otherwise that it's possible they could bounce right back and be normal. Whereas an elderly person with pneumonia is going to take a long time to recover afterwards. But some of these kids, if you could just blunt that cytokine storm, you're right. But the funny thing is they tried early, not funny, they tried early goal-directed therapy in kids, and they found out that loading kids up with fluids is the exact opposite effect in kids. It made them worse. So the FDA likes to say that kids are not small adults, and they definitely aren't. When it comes to sepsis, they respond very differently.
Yeah, that's interesting. I was reading that protocols were changed in COVID to intubate a patient, but put them on their belly in order
Right.
for them to increase like survivability by 13%. It was pretty large.
Yeah. And we were all discovering that in real time, weren't we? I mean, that was happening as we watched it. So people who criticize the CDC for not knowing when we were all learning at the same time is short-sighted.
That would mean the patient could speak as well, wouldn't it?
They're intubated. They don't speak anyway.
Oh, they're intubated anyway. Okay.
Yeah.
I have a question. In some of your discussion, you were talking about maybe having a drug approved when there were like 200 patients a year. What's the incentive for a pharmaceutical company to spend all that money developing a drug when there are only 200 possible users?
That's an issue. That's exactly it. But if you get orphan drug designation, you'll get exclusivity for marketing.
Right.
And then there's a number of things that hopefully, once you get the drug out there, it will often be found to do other things. But no, you're right. I mean, look at this treatment for there's a drug out there now that's a DNA that gets injected into the eye and cures blindness, and the injection costs $1 million. I think it's $1 million or $1 million per eye, and they're going to attempt to get their money back, but somebody figures out that getting your sight back is worth $1 million, and it so they play these games. And in our case, the price of this drug was priced pretty high. I was not real happy with it. They said, well, you calculate out the number of years that a patient will survive, and you base it on so much per year. And then even if they get out of the ICU, a couple days early, what's that worth? That's worth a few $1,000s right there. So they've got it figured out. Those guys, they play these games with costs and things. And it had nothing to do with mill rate. It had nothing to do with cost of actually making the drug. They just figured out, we've got to get our money back. We're, losing patents, and we just spent, $300-400 million. So we got to get that back first, and then we can start returning money to our shareholders.
Well, we're going on 8 o'clock. This went better than I thought. This is really interesting.
Oh, thanks.
It was, I just, being kind of an engineering physics kind of guy, I sometimes the medical stuff doesn't, but this is fascinating.
I'm a physicist, and I think it's fascinating. It's so important. And it's an example of the country who accepted risk to found a new nation on a dangerous continent, now afraid to accept the risk of driving more than 55. If risk could be accepted in medicines, pharmaceutical companies could manage. We'd have a chance.
You're absolutely right. But risk, even when people are willing to take the risk, they got a lawyer who's standing behind them willing to sue. Recover them.
I mean, I'm actually wondering, I was had some of these PD-1 blockers, Ipi and Nivo for cancer, and it turned out to be a good start, and then they switched to something else. But I mean, and clearly it was worth taking the risks. I mean, there's no doubt about it. But I mean, there's a case where, in fact, there's a whole list of risks, and the poor doc that was treating me, because I know a bit of statistics, what's the chance of this and what's the chance of that, and I'll get back to you tomorrow. said, yeah,
Yeah.
let's go ahead.
Well, cancer's a whole different ballgame. You know, not to be, casual about this, but the consequences of cancer are so bad that people are willing to take a lot of risks to get
Yeah.
treated. And the PD-1 inhibitors are just an amazing, because some of them have almost no side effects, and others, the side effects are dramatic and things like that. But on the opposite, the complete extreme of that is a treatment for diabetes, which can't have any side effects. You can't have any cardiovascular. You can't have any GI or liver. You know, you're going to take something for 30 years and you have diabetes. It's amazing how scrutinized the spectrum is. You know, a diabetes drug or something that's given to kids for a lifetime compared to a cancer drug. It's like, wow, it's like the Wild West for cancer drugs. And now we're getting better. I mean, the side effects are going down. The drugs are getting better. I know people who have completely cleared of life-threatening melanoma, and it is amazing. You know, so sometimes you hit, sometimes you.
Well, one more, not completely cleared, but doing a heck of a lot better than not a heck of a long time ago. I mean, it's really quite amazing.
Good. Yeah.
Tanner,
Yeah, the immune system. If we could harvest the immune system, I'm all for it.
Dan, what do you think the chances are in 10 years or 20 years or something that this particular molecule will emerge as useful just for something that, you won't benefit from probably, or obviously the company won't, but it will prove to be effective for something in medicine?
I think because we're first in class and the potency and the selectivity, I think it will be used for something. And my thoughts were, let's give it to the chemical company to sell the researchers, to get it out there. And I'm confident enough that nobody's going to find anything really weird with it or whatever. And let's find out what this molecule can do. Because there might be diseases out there, how do we know the inflammation around multiple sclerosis? We don't know. We know so little about these diseases that I would be all for, giving it a shot. And then, somebody will pick up a clinical trial. But the problem is there's no patent on it. So who's going to do a clinical trial on a drug that they're not going to make any money on? So that kind of breaks my heart, but that's the game.
All right, so Dan.
Thank you.
Good job.
Thank you, Dan.
Yeah.
Thank you.
Thank you.
Thanks, Dan.
Really well done.
That was awesome.
That was really good. Yeah.
Thanks a
Thanks
lot.
for your
Let's go to
patience.
sleep.
Good.
Tomorrow we get our COVID shot. So I'm already planning how lousy we're going to feel because this is number 2.
You New Yorkers, you're kind of aggravating me. I'm not able. I can't even get on a waiting list here in Bergen County.
Well, the Johnson & Johnson has just been approved. So that'll add extra medicine into the pot.
I hope. I'm hearing everybody I know is getting their first shot, second shot. I'm not even.
Right.
I'm getting my second one tomorrow morning.
Thank you. I need to be here.
Congratulations, Neil.
Never mind.
I
We're
feel
going to
great.
have to yet get eventually the third shot because of all the variants that are coming up now. I know that Moderna is working on one.
Oh yeah, they all
Yeah.
are. My son
They're gonna
is
have you
working
so
on
shot
one.
up that in the next few years, you're going to be like a pincushion.
It's going to be like the flu. Every year you're going to get
Every
your
year,
shot.
yeah.
Three.
You get 2 of them.
Yeah.
Bruce, let me thank you. Let me convey to Rick Schumann thanks for his great talk. I made notes from it. So I went to his YouTube and wrote myself some notes on it. It's that good.
Yeah.
I'd like to send him a copy if you tell me how.
Okay.
Okay.
Send me
So
an
you
email.
gotta reach me? Have any chance of reaching me by email or something like that?
Well, you can reach me. You got
How do I do it?
every time I send out a notice, they
It's
come
in
from
there.
me.
Okay, yeah, I'll do that. All right.
Respond to that.
Very good. Thank you all.
Yeah.
Thank you all.
Thanks
Take-
again, Dan.
Thank you. Thank
All
you.
right. Have a
That
good
was-
night.
Take care.
Thanks. Right.
Bruce, thanks
Okay.
for the diversity
Bye.
of talks. Thanks for the diversity of subjects, Bruce.
It is good.
It's,
Bruce.
it's,
Thank you.
it's come up organically.
Well, it's people care. This whole crowd is curious about everything.
Okay,
Good.
take care.
Thanks, everybody.
Bye.
Thanks, yeah.
Good night.
Good night.
Good night.